Good morning, good afternoon, good evening. Welcome to the APSA 2025 Plant Breeding Innovation webinar. Before the webinar starts, let me take 2 minutes to give you a short housekeeping rules. The participants are not allowed to open the microphone and camera. If you have any questions, if you have any technical issues, comments, or remarks, please put in the chat box. If you have any questions for the speakers, please put in the Q&A box. There is a like button in the Q&A box. If you find the question is interesting or you want to ask the same question, please just click the thumb up button. The questions have the most likes will be prioritized and answered by the speaker first. That's all for the short housekeeping rules. Let me invite Dr. Shwendra Bajaj, APSA Technical Advisor, to open the webinar. Shwendra, floor is yours.
Thank you, Shafong. Good day, everyone, and thank you for joining this APSA 2025 Plant Breeding Innovation webinar. We are really thrilled to have two speakers with us, and also we are very excited to see so much interest in listening to this webinar. This is the second of the webinar in our series. We had the first one last year, where we had discussed about the global regulation and status of genome editing in crops. And it's such a fast-moving area. I'm sure a lot of new things have happened in the last 1 year. So before I invite the speakers, I'd like to say a few words. Genome editing in our region has been adopted. The guidelines have been developing in many countries. Many countries have already adopted that. Some are in different stages of development. So today's speaker, through today's speakers, we will share about the genome editing guidelines in our region, and also what is the status of the guidelines in America, where it all started. So without further ado, I'd like to invite our first speaker, Michael Leader. He is the chair of our Plant Breeding Innovation Committee and also represents Bayer Scientific Affairs. Michael, all yours. Thank you so much.
Thank you very much, and it's an honor to have the opportunity to speak to you all today and to update you on some exciting bits that are happening in this space in Asia as well at the moment. I'll just see if I'm sharing my screen. Am I audible, and can you see my presentation?
Yes.
Yes, Michael, and
perfect.
Yes.
Perfect. I'll continue then. Yes, I have the honor of being nominated as the current chair of the Asia Pacific Seed Alliance's PBI, or Plant Breeding Innovation Standing Committee. It's a relatively new committee that's been around for the last couple of years. We've seen this issue becoming more and more important in the region and for regulatory officials as well to want to understand what's going on. So we've really set this up. What I want to talk to you about today is really, I guess, what's happened since the last time we updated the actual membership here at our Seed Congress last year, but also for those that may be new, that are publicly coming into this webinar, to get an update of where we are at in Asia as well and what's happened over the last few months. So for those that aren't aware, APSA's mission is to support sustainable agriculture through the production and trade of quality seeds for the world. It's been around for over 30 years and involves a number of seed companies, but also other groups as well that are interested in really bringing seed, which is the start of everything we do, to life in appropriate regulation and allowing food to get to people where and when they need it. At the moment, there are 495 active APSA members, so it has significant reach across the region, including East Asia and into the stand countries and Turkey also. But it also covers that we do have membership from outside, from the US, from many in Europe as well, and also from government seed agencies also that are interested in getting information and the most updated, I guess, advice on regulatory requirements requirements, but also technologies and other aspects as well. Obviously, most of the membership we're talking about are coming from significant seed production countries such as India and China as well. But it covers most of the countries in Asia also. The way that it's set up actually is that there are some standing committees that are working all the time on clear issues of policy work that we need to work on, including plant breeding innovation, but also phytosanitary information and requirements for seed movement through our International Trade and Quarantine Committee. But intellectual property and access and benefit sharing is also critical here as well. And then there are specific special interest groups for particular regions or industries, such as a vegetables group or field crops, or even the technologies associated with seed production. Of course, I'm just going to talk about plant breeding innovation or genome editing, or some people might talk about specific technologies like CRISPR, what it is, and just to briefly explain why we're interested in this new techniques that are coming through and where the innovation has led us today. Really, this is an extension of what current plant breeding or conventional plant breeding has been able to achieve for us over thousands of years, from when the initial corn to what we have today, significantly different thanks to conventional breeding practices. It is a molecular biology technique that allows us to get to precise areas in the genome and make changes to genes that we know may be of interest to us in certain pathways, like disease resistance or salt tolerance or other aspects as well. What we really like, and why breeding and breeders in particular are really keen about this, is that we can actually get these things to market a lot more quickly and take this variation and change through. It basically acts as a scissor to cut open a particular pathway and make changes that you want and close that pathway back up again, specifically into the varieties that you're interested in, which means you can bring it to market. You don't have to back cross and get out of all the unwanted traits that come from sexual reproduction. But you can actually target the pathway specifically that you want in an elite variety, bring it out to the farmers and continue giving them the yield they want within 4 years, as opposed to a lot longer. It can take up to 40 or 60 years if we're talking about trees or other things with longer generation times also. So this is why the seed industry is very interested in genome editing and what it can provide to variation and to change, and to allow us to really speed up what we have been doing in terms of innovation and evolution of our plant varieties to date. The problem is, because it does actually originate from a molecular biology technique, but can actually just create variation without actually leaving novel DNA or foreign DNA in there still are concerns about whether it should be treated as a genetically modified organism or not, the results of these particular techniques. So there has been still around the world debate about how to best regulate these techniques, unfortunately. There's non-harmonized policies. Every government seems to be taking their own time and policy thought pieces on how they're going to regulate this. Do they treat it as an identical to conventional crops that are coming to market, which means it can go through the same kind of pathway and get to market quickly? Or are they going to treat it as a genetically modified organism? As we know, it can take 16 years and over $100 million to get a GM crop to market because of all the significant regulatory hurdles that are put in their pathways at the moment. After 30 years of GMO technology, it's still taking longer to get to market than it did 30 years ago, which is quite extraordinary. But that is the kind of cost and information that regulatory hurdles can provide here. So depending on how governments are going to look at this, really depends on how quickly and whether this technology is going to be integrated and invested and available to farmers, and not only to significant crops like corn, but also to ones that are significant in specific countries as well, that may not have the big return on investment that companies need to put in order to make sure that they can afford to take the 16-year timeline as well. The Asia Pacific Seed Association does have a position paper on this, that we do believe that government policy should facilitate innovation on these advanced breeding tools, and that the latest plant products derived, from these tools should not be covered under the scope of genetically modified organisms if there's no foreign DNA present, right? So they should be looking at regulating the same as conventionally bred plants, if indeed the same result can be obtained through chemical mutagenesis, sexually compatible breeding, or other aspects of mutational variation that has been going on at the moment. So currently in Asia, it's still a little bit all over the shop, we would say. In English, there are a number of non-harmonized requirements and regulations and data requirements that are needed. But there is a move in the region, as you can see, to consider that some of the results of these techniques are not genetically modified organisms. Australia allows specific SGN1 or very, very limited scope to allow for cultivation and commercial release. We're seeing policy developments in other countries as well. Thailand and Singapore have recently allowed for exemptions for no foreign DNA present. They don't need to be treated as genetically modified organisms. Philippines and Japan have had significant frameworks in place for some time, and probably the oldest in the region. India and Bangladesh are following as well, and have recently, I think 3 years ago, did put in place some regulatory exemptions as well. But again, the information required, the kind of edits that are exempted are all slightly different in all of these schemes as well. It is non-harmonized. China is still really considering them to be genetically modified organisms, although there may be some simplified assessments depending on what the government decides, whether there are reduced risks as well. So it's a more difficult regime to navigate there, to work out. There is legislation and debate happening around the region though, so this is good. But now, what I want to talk about is what's happening since February as well, and what we've been trying to do in the Asia Pacific Seed Association to harmonize this, but also what is actually, what are the new exciting developments that have happened in the last 6 months or so, really. What we did do is set up our first regional plant breeding innovation expert consultation, which is something that we've been doing for some time actually in phytosanitary world, and also in I guess the access and benefit sharing plant variety protection space. But we had our first consultation where we invited a number of countries, governments and policy officials, academic experts from around the region together to discuss where we're up to with this technology, what the future potentially holds for this technology, and discuss what actually needs to happen to make sure that we can take advantage of it in the region also. We had more than 14 economies actually turn up to this particular meeting from the region, which is very, very positive for the first time that we've held this. And of course, we had international seed industry representatives as well as people from APEC as well, which has really been quite good. The reason why we found that this model works actually is that it focuses on building trust. People get to know each other, who's actually working in seed production. The government, even the different governments working in this field, get to actually trust each other and understand what's going on. They actually get to ask the questions in a trusted, closed environment on things that might be troubling them or that they don't fully understand or grasp on the technology also. So it is a relaxed and informal setting. And because we hope to have this keep going for a year-on-year basis, it gives an opportunity to track country reports. They provide reports on what's happening. We can see and track progress and help to explain any particular, again, capacity gaps that might exist as well. Partnerships have always been really important to the Asia Pacific Seed Alliance, and we will continue doing this. And this provides us the opportunity to do it in this space. We've been doing it very well in the phytosanitary space and in access and benefit sharing. But this is a really new key critical area to be able to do this as well. We like to think that we did get some, this actually created momentum for some governments to continue and progress in this field also with their regulatory frameworks. So let me now maybe spend a little bit of time just trying to explain what's happened since then, right? And the first one I want to talk about is Australia and New Zealand. On September the 2nd this year, we actually saw a new definition for genetically modified food introduced into the Food Standards Code. This code regulates the safety, health and safety of food products both in Australia and New Zealand. It currently does have a regulatory framework for GMOs, or food derived from genetically modified organisms, but that definition was changed to mean food from an organism that contains novel DNA. Now, this was pretty exciting because that actually meant, okay, that means something from a new organism, or if it can be done through conventional breeding practices, it's not a genetically modified food. It doesn't have to go through that pre-market assessment that a GMO food might do. Fazan's actually told us at the APSA presentation or expert consultation, they were doing this because they wanted to update and provide a clear and objective definition that actually captures modern technology and techniques, something that was easy to comply with and to enforce, and that was consistent as well. So what they did present was that they were really moving from a process-based part. Was it done through molecular breeding techniques, but more actually looking at an outcome-based definition? Does the food from that product actually contain novel DNA, right? Does it actually, in the end, have a new gene from a non-cross compatible species put into it? Has it created something that doesn't exist already in nature, or that can't be done through conventional breeding practices? So they did capture as novel DNA. Obviously, if you insert DNA from something, a bacteria, into a plant, something that can't actually happen naturally, that would still be considered a genetically modified food, right? If you arrange the DNA in some way that couldn't happen naturally, that will also be captured. Also, if you synthesize DNA that cannot exist, that doesn't exist, that may also be captured. But if you insert DNA or make a change that can result from natural processes, such as a transposon change or from sexually compatible breeding, that is no longer captured as novel DNA and will no longer be regulated as a GM food. In fact, it's up to the developer to make their own decision on this. It's a self-determination process as well. It gives you a lot of flexibility to really work on this and to make and speed to be able to bring the results of your innovation to market here, if it's a result of genome editing and falls out of that novel DNA definition. So that's happened, and that will be in place from the 2nd. It's been in place since the 2nd of September as well. We also see that Indonesia presented a new regulatory framework and updated their particular biosafety clearinghouse information post the expert consultation as well, to make it clear that applications to exempt genome edited plants from regulation as a GMO was also something that was allowed now. You need to make a presentation, right, to the Biosafety Committee, but if there's no new genetic combination, or in fact, the new genetic combination was from inside the taxa, which they're still working on the guidelines as to what this means, but some of the information that were provided with means that things from cross-compatible species will be exempt as well. So as long as you make a representation, an application to this particular committee, they will do a quick assessment and provide the information about whether it's a GMO or not. This is really exciting because Indonesia does have a lot of work that it's doing there as well in the genome editing space, particularly for plants, covering rice, covering and other aspects as well. I think corn. I think they were looking at certain vegetables also. If they can get this to market and provide the benefits to the country there, that will be really good. So they are accepting applications. They also presented what actually information is required in order to get the decision as well. It's minimal information, really, in terms of the molecular data you need to provide. You just need to provide information that there are no foreign genes at the site that you want to edit. This is probably the most simple type of information you can provide there, as well as information on what you intend to do, what's the intended effect, how you made that change. This follows a lot the Philippines, I guess, information network and data information that's required as well. Of course, if there's a novel gene and you're changing the way or oil content or some of the other becomes a novel food, then you may need to look at it a little bit differently. But to make changes that can be done to conventional breeding as well, this is the type of information that needs to require. So Indonesia is really somewhere has progressed since that last map that I showed you, and is accepting applications now for genome edited products to be treated not as GMOs. I will, and I know I'm running out of time now, but there were developments in Vietnam as well. Currently, you saw the genome edited products are still regulated as GMOs because of the definition in their biodiversity law. But we do know that in June this year, the Prime Minister issued a list of strategic technologies they want to progress and take advantage of in Vietnam, and that included genome editing, not just in medicine, but in agriculture, which is a significant shift in policy there. We were also informed at the APSA consultation that they're looking to draft a new GMO definition right now that will hopefully allow for exemption of genome edited products and a regulatory framework to allow that to happen. So we're hoping that will be that will happen by the end of the year. There will be a new definition in the law, and then we can work on the secondary legislation, and they can work on information requirements that may be provided there, the kind of steps, timelines, and things that might be required in order to exempt a GE product. But certainly, I think that legislation should be in Parliament by the end of the year. Fingers crossed there, but that is a positive movement in Vietnam too. We heard that Malaysia has recently actually looked at their research and development phase in the laboratory and made changes as well. So you now can be exempted from doing full-on biosafety conditions to do research in Malaysia if you're dealing with a really limited scope of genome edited products. We hope that they will increase that as they get more familiar with it, but that does free up the local research teams to start not having to actually require as many confined requirements or conditions as possible. We hope that they will then extend that soon. They did say they were looking at now trying to extend that to field or breeding trials out in the environment, so that these people can actually see how their ideas are working in the natural environment. This will be critical to see if it actually has a fit for Malaysia or for globally as well, right? We're also looking at if you have something in the US or Brazil or Vietnam, can it actually be imported as a genome edited product as well? They're still debating that, but we hope to see policy in that space as well, potentially in 2026 too. So Malaysia is also moving ahead, although at a slower pace. But not only that, we're seeing a lot of other developments, okay, in Asia. We have 22 approvals of exemptions for genome edited products at the moment. 10 in China, 6 in Japan, 4 in the Philippines, and 2 in India as well. 2 genome edited rice in India that are now have variety registration approvals. We're seeing it across different crops, 7 crops, 3 fish species as well. I believe there's an application for a GE rice in Indonesia as well that hopefully is a long way through that exemption process, and we might hear some positive news there soon as well. In Chinese Taipei, the genome editing landscape is going to be reviewed, we heard, before the end of the year, and hopefully that will follow a lot of the movement we've seen in other countries as well. In Japan, they've had a scientific expert meeting and agreed that cisgenesis, or where you don't have novel DNA that comes into the resultant, to a resultant product, is equivalent to natural occurrences for the purpose of GE regulations. So hopefully there will be moves to update and future-proof their regulation with this next year also. Hopefully there'll be another submission for an increased grain size rice in the Philippines before the end of the year too. We know that Pakistan is also now developed a biotechnology policy, or a draft one, that does follow some of what Philippines, Thailand, Singapore, and Indonesia have done. We hope to see that passes through cabinet before the end of the year as well, which will lead to the development of regulation. And New Zealand and Australia are both updating their cultivation requirements to exempt more broad expanse of genome edited products as well from regulation of GMOs, and we should see progress on that in 2026 as well. So really there is a lot of exciting parts in this, and I believe that APSA has been a key focal point to help provide capacity building information to governments thinking about this, showcasing what's been done globally, but also providing a forum for people to share ideas as well. So if there's anything or key message I want to leave you with today, is that a lot is happening in APAC in this space, and please always reach out to the APSA Secretariat or to committee members if you want any further information, or that we can help with any updates or capacity building events that you might have in your country as well. With that, I'll stop sharing and say thank you, and hand it back to Shivendra.
Thank you, Michael. Very useful presentation. Good updates of the latest status in our region. We already have a few questions, but I guess we'll take the questions at the end. I now invite Fan Li Chow from American Seed Trade Association to update us about the regulations in America. Fan Li, all yours.
Thank you, Shivendra. Can you hear me?
Yes, thank
I'm
you.
going to share my screen. Just give me a minute, please. Please let me know when you can see my screen. Can you see my screen?
Not yet.
Okay. It
But
might take a
coming.
minute.
Yeah.
I'm on the other side of the world.
Yes, we can see now. Thank you.
Okay. Thank you to the APSA PBI committee for the invitation. I'm going to give everyone an update on the PBI regulatory and product development in the US. So before I start, just a little note about ASTA. We are a member of APSA. ASTA stands for the American Seed Trade Association. We are one of the oldest trade associations in the US, consisting of around 700 members that's involved in plant breeding across the different species. We like to say A to Z, alfalfa to zucchini. And most of these are actually small businesses that has less than $15 million in sales. So our mission at ASTA is to be the voice of the U.S. seed industry, but advocating for a policy environment in the United States that really supports agriculture and resilient economic development. We also represent our members at the international level, at the national level, and at the state level. So by uniting our members, we do quite a lot of communication to the public and also communication to our members around science and policy that is impactful for our industry. So ASTA, we are I guess a node in a global network of seed partnerships. So we partner with international, regional, and national seed associations. So we are partners with APSA, but also operating under the umbrella of the International Seed Association. And the reason for that is, from our members' perspective, a seed production, the most efficient way that our members have uses to develop the best variety in the most efficient manner to our farmers, both in the US and globally, is through a global product development, product production supply process. So this is an example of how a tomato seed may move around the world from R&D to production, to distribution, to cleaning. So it can go through 6 or 7 different countries, crossing international borders, before it reaches into the hands of a farmer. And from our perspective, like all of our seeds that we produce are subjected to a number of regulations, right? And these regulations, for the most part, are not based on any technology. So we create seeds to develop seeds, breed seeds through a number of different technologies like conventional breeding, back crossing, molecular assisted. All of these seeds and the products that derive from these seeds are regulated. But we do have one set of regulation that is very specifically tied to a technology, and that is regulations that apply to plants derived from biotechnology or GMO regulation. So that is what we're here to talk about today, is now that, as Michael mentioned, I guess 10 years ago, we started seeing genome editing being applied in plant breeding. And the biggest question was, for seeds that are developed using genome editing, are they going to be treated like GMOs under the GMO regulation? And as Michael mentioned, this is quite important for the broader seed industry. So from an APSA perspective, most of our members do not use GMO breeding technologies, right? Because the cost of both US regulatory compliance, but also global regulatory compliance is really out of reach for many of our vegetable seed companies. So when genome editing came on board, this really powerful tool, many of our companies were really concerned about how the products would be regulated. Because if these products are going to be regulated, GMOs, then it really priced many of our companies out of the GMO space, right? So that's how APSA kind of got involved in both domestic advocacy from a regulatory perspective, but also international advocacy. So I'm going to just give everybody kind of an overview of how the US government regulates products of biotechnology. And what we did is, in the US, we established a US coordinated framework for regulation of biotechnology, so shorthand CF. This was established in 1986 when the first BT crops came onto the commercial landscape. And what the US did is quite different from a lot of the rest of the world. We did not pass any new laws. So in many countries, there are GMO legislations, GMO acts, GMO laws. We did not do that. We actually used existing laws and looked at the intended use of these products that would be developed using biotechnology to see how will we regulate them. So we're regulating them based on their intended use. For example, if your biotech product is going to be used as food or feed, so food for humans or feed for animals, then you would be regulated by FDA, because FDA regulates all foods and feed. If you're intended to use this product as like a crop protection, so for example, BT toxin incorporated into a corn plant, then this is intended as a pesticidal use. So EPA regulates pesticides. So we will use the same type of regulations or same legislation to regulate plant incorporated protectant, right? And for USDA, what they regulate is for any plants that goes into the commercial planting. So if your biotech plant is going to be intended for planting, then it will be regulated by USDA. So each of these agencies have different legislative or statutory authority, and they apply them to the products of biotechnology. So in our case, biotech plants. So when genome editing came on board to be used in plant breeding, the question was, how would this coordinated framework view products derived from plant breeding innovations such as genome editing? And this has been an evolving conversation in the US government for the last 10, 12 years. So under each of these federal agencies, USDA, APHIS, FDA, and EPA, they look at things very differently, right? So the first line is, what do they regulate? So under USDA, they regulate certain genetically engineered organism under the regulation 7 CFR Part 340. That's just the name of our regulation. And the question that USDA would ask is this plant safe to grow? And they have this process called MI regulated process, where as a plant developer or a plant breeder, you can write into the US USDA and say, I have created a plant using genome editing, and I'll show you an example of this. I believe I do not fit under your regulatory authority. Can you confirm? So this is called the MI regulated process. Between 2020 and 2024, USDA also had a confirmation process where they had certain categories of exemptions. So if a plant had certain modifications, it was exempted from regulation. And as a developer, you can write to USDA and says, I've created this plant. I believe I'm exempted under certain categories that were stipulated by USDA. Can you confirm my finding? And this was called the confirmation of exemption. So under FDA regulates all food, all feed, regardless how they are produced. In 1992, they put out a policy statement talking about how their regulatory statute for all food and feed could apply to plants or food derived from plants that are created using biotechnology. So when genome editing came on board, in I think last year, they put out a guidance. It's like, this is how we would view food derived from plants produced using genome editing. And what FDA set up is a pre-market meeting. So before you put the product on the marketplace, you can go and have a meeting with FDA, explain the intended use and the intended modification of your plant, and just having a discussion with FDA to assure FDA that your plant does not present any novel food or feed safety. So for traditional, for transgenic plants, for GMO plants, FDA has a pre-market consultation, which is a lot more extensive in the amount of information they review as compared to genome edited plants. At EPA, their concern is when a plant is used with a pesticidal intent, so pesticidal substances like BT, and they have what they call a rule for plant incorporated protectants. So these are pesticidal substances or substances with a pesticidal intent that's incorporated into a plant. The most common example is BT corn. Okay. So for EPA, they recognize that you can create plant incorporated protectant through conventional breeding, right? We can breed plants that are resistant to viruses, to fungus. So they have an exemption for PIP that's created through conventional breeding, recognizing that fact. Understanding that we would now use biotechnology and plant breeding innovation like genome editing to create conventionally like PIPs. EPA also in their 2023 rule created a category of exemptions for plant incorporated protectants, PIPs, that's created through genetic engineering derived from sexually compatible plants. So they also exempt PIPs that are loss of function, right? Because there's no novel genetic modification with genetic insertion. So all of these are different ways of saying that if a plant could have been done through conventional breeding, then it is exempt from the regulation, from the biotechnology regulation that each of these agencies promulgated, right? So I'm going to talk mostly about USDA because USDA is the agency that will see most of all the plants, right? So any plant that is modified, USDA has authorization over because they're concerned about if a plant is safe to grow. If your plant is not going to go into the food supply or the feed supply, you do not have to go to FDA. And if your plant does not have a plant incorporated protectant, you don't necessarily have to go to EPA. So USDA will see most of the modified plants. So I'm going to focus on that. So the regulatory approach that USDA used, the MI regulated process, the AIR, the AIR process, is their concern from USDA's perspective is the plant safe to grow? So can the plant be a source of plant risk? So does it create risk for agriculture? And they have a regulated article definition. So anything that fits under this regulated article definition is then regulated. And that regulated article definition is focused on the characteristic of the final organism and whether that final organism has a vector or sequence from a plant pest, right? And USDA has a list of what they consider plant pests. So this is very compatible or comparable to this novel DNA or foreign DNA trigger that other country uses, right? Because other countries has a GMO regulation or GMO law, USDA has a plant pest regulation, so they're concerned about foreign DNA that's of a plant pest origin. So the MI regulated process asks, does the final product contain a plant pest sequence? So USDA started this process in 2011, and this is where we started with and since 2011, we've had USDA have issued over 200 letters, MI regulated letters, and you can find them online, and I'll pull up the QR code for you to go to the online database. So one of the first letter that was issued in 2012 was a letter to Dow AgroScience for the use of zinc finger nucleases in soybeans. And what Dow AgroScience presented to USDA was four main points. One, that zinc finger DNA constructs were used to modify a plant, and the zinc finger DNA construct did not contain any plant pests or pathogenic DNA, or and the plant that was modified using the zinc finger nuclease contained no recombinant DNA or foreign DNA in the final product. The letter also says the final product is indistinguishable to non-regulated mutagenesis products, so products that are derived from conventional mutagenesis. And the zinc finger is designed to target very specific gene of interest. So, and then it kind of described what the modification was, which was a deletion of the IPIG gene, right? So this is all the information that Dow AgroScience provided to USDA. And Dow AgroScience's letter says, our plant does not fit under the regulated article definition and therefore is not subjected to the biotechnology regulation of USDA. Oops. And so USDA issued a letter in response that says, based on the information you provided, we agree with you that your corn plant that contains a deletion at this gene using zinc fingers is not considered a regulated article, so not regulated. So am I regulated? And the answer is no, you're not regulated. That's where we started. One gene, one modification in one crop, right? This is where we're going. So USDA now had over 10 years of experience issuing these MI regulated letters. So this is a letter that was just issued in July 2025 to the Trade Company, which is a genome editing company that is editing products for GDM, one of the biggest soybean germplasm companies. And so what the Trade Company provided to USDA is a description of an approach to edit soybeans to improve yield, right? So what the Trade Company letter described is that we are going to edit many, many lines of soybeans to effectuate yield phenotype, and they listed the different types of yield phenotype. It's been redacted for confidential business information, but you kind of can see that that is what they named. It provided an example of a vector. It provided a description of a scientific methodology to confirm that the genetic change occurred, and it described a scientific method to confirm that there's going to be an absence of plant pests and foreign DNA in the final product. It listed 600 gene targets, which is also redacted for CBI, but it's very clear. It listed 600 gene targets, right? And it did not list every single guide RNA. It did not list every single vector. It just merely described the approach that they're going to take. It described the gene target they are aiming to hit, and it described how they're going to make sure that the genetic modification is there and that there is no foreign DNA or plant pest DNA in the final product. The USDA response to trade companies' "Am I regulated?" letter is that it kind of talked through what was described by the letter, that the trade company is going to use a CRISPR-Cas system to induce indels in 600 targeted soybean gene sequence related to yield improvement. Right? It's all confidential business information. It's saying that your method is going to create gene edits using targeted DNA breaks. And the letter is stating that foreign DNA, the lack of foreign DNA, including plant pest DNA, would be absent in the final product, and that would be confirmed by a method that is described, right? So this method is described to the agency. It's CBI in the letter we can see. So based on the representation that the trade company provided to USDA says, we do not consider your modified soybean lines to be regulated pursuant to 7 CFR Part 340. That's the biotech regulation, right? So this is where USDA is going, moving away from am I regulated determination for a single gene, for a single modification, to multiple genes, multiple modifications, recognizing that the final product may not contain all 600 different target sequences, but recognizing that the regulatory statutory authorities is only for products that contain a plant pest or foreign DNA, and these do not or will not, right? So this is really a way for USDA to shift away from the GMO mindset towards how genome editing would be used in plant breeding. So the GMO mindset was very much one modification, insertion of one gene, and it effectuate one trait development, right? One gene, one phenotype, event by event, actually one insertion of that gene into a germplasm, resulting in one phenotype. From a regulatory perspective and from a GMO perspective, and the way it was used, a regulator and a plant breeder would be only submitting a handful of candidates, right? So it's going to very small amount of events that's approaching the regulatory agencies. From a gene editing perspective, we can certainly do that. We can certainly insert or modify or perturb one gene to effectuate one phenotypic expression. So that is kind of what Dow AgriScience proposed. But we can also use genome editing to create a variety of genetic variations very early on in the plant breeding process. And then use that from a selection, then use those variations to be selected upon, right? So in this way, this use of genome editing as a plant breeding tool, as a tool to create genetic diversity, would mean that there's a greater number of modifications, a greater number of combinations of modifications, and potentially a greater number of these things coming into the regulatory agency. Because from a plant breeder's perspective, I would like to put these things into field trials or into the field, and I would like to know that these are not regulated, so I do not have to then have that regulatory compliance for a GMO. So then the approach or the amount of information, the amount of applications, if you will, that's going to a regulatory agency can be exponentially increased. And this is the reason I think USDA, as it's moving from 2011 to now is really changing how they are looking at these MI regulated letters. Right. So this is just a kind of a slide that I borrowed from our colleagues at Bayer that really looks at the differences between how genome editing could be used in a plant breeding process versus how GMOs or introgression of a specific trait are used. And this is from a gene editing perspective, we can actually be integrated into the plant breeding process much further upstream of selection. And this creation of genetic diversity in elite genomes is what's creating the kind of the numbers that the trait company is approaching USDA was. They don't know out of that 600 candidates which one are going to be, or which combination of one is going to be the one they take to a commercial space, but they would like the opportunity to test these in the field and to have that early decision that this is not a GMO is really, really useful from a R&D and product development pipeline perspective. So just to give you an idea of what kind of plants that USDA have reviewed, this is kind of, you can scan these QR codes and we will be sharing these presentations after the conference. You can just go directly to the website, the MI regulated website, and see all the letters that was submitted to USDA and USDA's responses. Even though many of them are redacted for confidential business information, you can kind of pick out what the modification, the goals of these modifications are, and USDA's answers. So you can see the MI regulator letters, the USDA confirmation exemptions, and then FDA's pre-market meeting decisions that's online. So you can see there's a diversity of crops that's been reviewed, anything from almonds to tomatoes, and of course, corn and soybeans is in there as well. But this is really the fact that these products are considered not GMO from a regulatory perspective really opens up the opportunity for a diversity of developers to be involved in this space. So just as a closing summary, I want to just kind of highlight a couple of things. If we look at the R&D pipeline, the pipeline is robust. We're seeing a diversity of genome editing being used in different types of crops, from row crops to vegetables to fruits to trees. We're also seeing a diversity in the type of phenotype of the traits that's being modified. So we're looking at nutritional traits. We see nutritional traits. We see abiotic, biotic, but as well as consumer-friendly traits like seedless blackberries, seedless cherries. We're also seeing a diversity in the type of developers that's using this technology for crop improvement. We're seeing big companies, as always, but we're also seeing startup companies that are partnering, that are using their technology platform to partner with germplasm companies. And we're seeing university in this space as well. From a regulatory perspective, as Michael pointed out, it's positive. It's definitely positive. There's growing international alignment that genome edited plants are distinct from GMO plants from a regulatory perspective. And there's a lot of regional regulatory cooperation, especially in South America. There is a official collaboration between Argentina and Brazil, and there's also looking to expand that collaboration to the CAS countries and beyond the South America region. And I think this is the reason for that is because a lot of these genome edited product development is very focused on domestic economic opportunities and also regional economic opportunities. And so the incentives and economics incentives are slightly different than traditional GMO row crops. But of course, there are challenges ahead, and those challenges also create opportunities. There are, as Michael mentioned, regulatory schemes that are still unclear in many countries and many major markets, right? Looking at South Korea, China, the EU, those markets still have not determined what they have determined that genome-edited plants are GMOs, and that creates difficulty for a product that needs to move internationally from both a seed production and development perspective, but also from a commodity movement perspective. For countries that already have regulatory implementations for GMO and gene editing to distinguish between the two, there's some differences in the implementation, right? There's some countries allow you to have early consultation. So if a product is in the conceptual stage, they allow you to go in there and say, conceptually, we're doing this. Is it conceptually a GMO or not GMO? And this is really important for small companies, for startup companies that need that assertion, that certainty that their product development is not going to be a GMO to continue fundraise and continuing move down that product development pipeline. The information that's required to make that determination of whether a product is GM or not subjected to the GM regulation or not is also different. You can see that in the US, the data requirement is very, very small because all they're interested to know is whether there's a foreign DNA or plant pest DNA in the final product. They're not concerned about really molecular characterization or off targets because that's not their regulatory authority. Two, there's different timelines. So in the US, it could take a couple months for that determination to occur. In Canada, it could take 2 weeks. So that time differences is also really concerning for companies that are really trying to move their products through the commercial pipeline. And finally, we need to kind of think through that genome editing is it's not at a standstill, right? They are using this technology to really do a lot of really exciting plant breeding modifications. And we need to really think about how our regulatory frameworks can keep up with the science of plant breeding innovation and try to move away from our GMO mindset of looking at these product development from an event by event trait phenotype perspective, right? Where really plant breeders are really trying to use genome editing in a very different way than we use traditional GM technologies. So with that, I would like to thank the organizers and the attendees for your attention, and I'd be happy to take any questions, and I'll stop sharing now.
Thank you so much, Fanley. Very useful update about what's happening in the US. We do have a few questions for both you and Michael, and I'll start reading from the top. So obviously, first few questions, perhaps Michael can take. So Ram Mohan has a query about what is the scope of novel DNA definition? Any other region or country aligned with this? So I think it's pretty broad question, open to both you and Michael and Fanley.
It is a pretty broad question indeed. I think every piece of legislation is slightly different in the way that it interprets things. The interesting thing about novel DNA, a lot of countries in the rest of Asia are using the term foreign DNA, or sometimes we saw Indonesia use the word taxa. So some debate about what all that means will be really important. But I think generally the view of some of the more recent regulatory frameworks that are coming out is that really it's about the outcome. Is there any foreign DNA or novel DNA in there from a non-crossable species? If not, if it can be juiced the same way through conventional breeding practices, how we normally have been bringing plants to market now, that it shouldn't be regulated. And that's really the important piece. And I think Fanley has even said, some countries in Latin America and US are going even further than that and really looking at creating variation and using this as a tool, as we have previously with chemical mutagenesis or X-rays or radiation, those kind of things, that it's even, or looking for somewhere outside for a wild type to bring into your breeding line. These kind of things are really the kind of things that these frameworks are trying to capture now, and we really need to start thinking about how to harmonize that and bring that future-looking mindset to Asia as well.
Yeah, so Michael, I was actually on my phone looking up the novel DNA definition that Fazan put forth because it's really very clear and I can read it, but I can
Yeah, I
figure.
didn't want to do that here. I had it in the slide deck. I'm like,
Yeah,
oh,
I think if
go, go.
you want to share it, we can add it to our slide deck and share it with the group, and that'll probably be the easiest way to do, because I think Fazan has the most clear definition of novel DNA, and it's very much based on plant breeding, right? What we have done from a plant breeding perspective and what we cannot do from a conventional plant breeding perspective. And they designed their novel DNA definition based on that. So we can share that in our, Michael and I will add it to our slide deck so you guys have that. But a lot of countries are so it's actually quite interesting, because I just came from a meeting in South America where they had a PBI consultation, kind of like APSA for the South American regulators. And they have slightly different definitions of novel DNA or novel combinations. But what they did was they did 3 case studies of actual potential uses of genome editing. And they had a closed door discussion just with the regulators. And then we came back together with regulator and private sector about how they would interrogate their own regulatory processes with these 3 cases and see if they come up with the same determination of GM or not GM. And it was interesting in like there was 14 countries present. In all 3 case studies, they all arrive at the same determination of not GMO, even though their regulations are slightly different in the novel DNA definition. So I think, we are somewhat in the US bounded by our statutory authority, but there are ways for us to come to the same answer, even though our sausage making may be slightly different. So I think that from a regulatory from a private sector perspective is super important, that the definition may be slightly different, but we find a way to come to the same conclusion at the end, so that there is alignment in the decision, the final decision across different jurisdictions.
Thank you both. Ankoro has a very interesting question. Do the current global regulations consider physiological aspects of plant that may be altered by genome editing? I mean, and the examples are given like changes in respiration, flowering, or senescence. They are not always apparent in initial phenotype. So yeah, so again, question open to both.
I don't know, Michael, I can start, but I think, we get so caught up talking about genome editing, and we forget that whatever we do to the plant from a genome editing perspective, it still has to go through the plant breeding process, meaning testing, selection, multi-year field trials, right? So I think this is where genome editing is merely a tool in a very, very complicated and rigorous plant breeding process. So our expectation is that we're still going to treat these plants like any other plants. We're going to be looking at quality, performance, all of those things. So I think, the idea is that they're still going to be treated like conventional breeding and conventional breeding processes. Right, Michael, I don't know if you have anything to add to that from a company perspective.
No, I think, we've been dealing with things that haven't existed in our plants for centuries anyway. If you look at what the original corn is, did it have a kernel? No, I think, for some things that we don't see now, they may still exist in a wild species that we can bring into what we want. It could happen naturally through a natural mutation. I think the carrot used to be purple, except for a point mutation that made it orange, and then we wanted it. So I think some things that don't necessarily happen now can certainly be obtained through natural breeding, but also through genome editing as well, whether it be short stature corn, whether it's something that a longer root system, a disease resistance, or salt tolerance as well. I think some of those aspects are all being worked on. Drought resistance, all of that is being worked on in some labs in genome editing space right now. So I think the answer is yes, really.
Okay. That's, I think, next finish for Michael. It's about Indonesia regulation. Rahayu has a question about, so in Indonesia, it's stated that within some taxa could be considered non-GMO. Okay, how do we counter with consumer concerns about allergenic reactions And it's a question about some religious issues as well. I mean, I think the overall question is about, can we still maintain the GM, the labeling part as in GM, if I understood question correctly.
I think they're still, Indonesia is still regulating GMOs and their labeling and those kind of things, but things that can be done through traditional breeding, is what they're actually going to now start exempting from GE. And of course, people talk about the safety aspect, but there is a novel DNA. They actually make it clear there is a safety assessment for novel foods. If you're changing, from a normal oil content, or you're doing a high increased oil, or producing vitamin D coming out of a tomato or something like that. If it's a novel food under the regulation, that will still undergo some kind of regulatory process. Food is being regulated anyway, regardless if it's GM or not. So that aspect's still there, so I don't think they need to concern themselves about that piece, if that makes sense.
Yeah, thank you. Hieronima from BPI has a question, actually two questions. If there have been cases where the three US regulatory agencies have arrived at different conclusions in their evaluations.
No. So the three agencies actually works really close together to make sure that their timeline for decision making is aligned, because that is the last thing they want to do is for USDA to say, go ahead, you're fine to plant. And then FDA says, we may have a question about your product. So the agencies, that's why it's called the coordinated framework. They do their very best to coordinate when they're open. Our government's closed right now, so there's no coordination. But yeah, we had a case back in the day where they actually split their food and feed authorization, and that created some difficulty. So we don't even do that anymore. So everything is tried to be as lined up as possible.
She has another question about the phenotype. Is the resulting phenotype also considered part of evaluation? I mean, if the trait is considered, if the trait confers resistance to a certain disease, is it necessary to establish the degree of resistance in the genome-edited product?
So, and from an EPA perspective, like they are not, what they're doing right now, what USDA is doing, what EPA is doing is not a risk assessment. What all these countries are doing is not a risk assessment. It's an assessment of whether it's in or out of regulatory scope, right? So if it's within the GMO regulatory scope, then it will go through a risk assessment. If it's outside the regulatory scope of the GMO regulation, then it's treated like a conventional product. So however those conventional products are regulated by all these other regulations we have, and that's how it's done. So in the US, actually, we don't have a varietal registration system like in a lot of countries, right? We are very much market driven. If your product is horrible, nobody will buy it. You will not survive in this US marketplace. So there's a lot of I guess industry governance that makes sure that you are a responsible corporate player in this space. So it's very different in the US, right? We have a lot of litigation here, and that may help corporate responsibility, but that is our tradition here.
Yeah. Naomi has a policy question. How do the approaches by the USDA bring opportunities for future-proofing policy developments for the genome-edited applications in plants in the Asian countries?
I think there is, I don't know, Michael, you can talk about this too, but I think from US perspective, like the first to go is not always the best, right? Because you make a lot of not necessarily mistakes. You go through learning, growing pains, and you can see USDA's and my regulator letters, the amount of information they started out with, and they just get inundated with the number of applications that's coming, and USDA trying to really figure out what is our regulatory authority here, and what is what are we looking at? So I think from countries, not just necessarily in Asia, but in other places that are just starting to develop their regulatory policy, there's lots of good lessons learned from the different countries. And even in South America, right? Argentina is put out their framework in 2018, and now they are going to put out a new guidance because they've kind of figured out like, okay, we didn't need to do that. So I think there is lots of lessons learned from countries that have done this, and this is where the APSA PBI consultation is really useful for that conversation to occur.
No, I think you're spot on, Fanley, and I think we have been trying to do that through ABSO and explain what's happening in the way that companies and want to use this technology now, not just to bring an edit by edit or a product to market, but to actually do the variation and the checks at an earlier stage. Just means that we need to start thinking, those that are developing their legislation now, say Vietnam, say some countries, Korea has a bill in front of Parliament, others need to think, do we actually want to be doing edit by edit valuation of this? The resources will be intensive when the concept is to try and get things out in the field, try and work and see what actually breeds the best valuation we want, particularly in the different climates and environments we want to be growing plants in. So thinking about that will be really critical, and to try and I guess open up the innovation that we've got going on in Asia, maybe some harmonization of thinking about this and information requirements at an ASEAN or at an Asian level will be really helpful to give that certainty and speed of innovation coming to the growers in this region too.
Yeah. Next is a very interesting question. It's a long question. Let me try to summarize it. So in the case of vegetative plants or asexual reproductive plants, how is the foreign DNA removed? How are the remnants of vector, especially agrobacterium, is removed or eliminated? It's a technical question.
This is a really tough question, and this is something that I think APSA members has been grappling with, right? Because like if we're talking about bananas, potatoes, it is really, really difficult. I mean, things are getting better, right? They're doing induced there are different technologies. They're trying to make this easier. But for us, at USDA, they've been really thinking about this question, right? If you're using a disarmed agrobacterium to do the transformation, what is the risk, right? Like, is there a risk? And in their rule from 2020, they have says disarmed agrobacterium is not a plant pest risk. So they actually exempted, they were trying to exempt that. That got kind of defeated by an NGO filing at the courts. But that is something that everyone is thinking about, is at the end of the day, what risk does it present? And perhaps we are a little bit handcuffed by some of our regulatory triggers that is going to create difficulties for vegetatively propagated plants, unfortunately.
I think Osman probably responded to that. I'm not so sure. He just made a comment. Regulatory determination versus regulatory risk assessment. And that's what he said. Anyway, well, this is a question which is slightly out of the scope, but invariably asked, even in genome editing meetings. Some people believe that GMO is not beneficial for environment. Is it true?
GMO or genome editing are we talking about here?
GMO.
Well, I mean, we've been, for 30 years, we've been having genetically modified organisms being grown in the environment. They undergo, numerous safety assessments. They produce, millions of yield increases. The amount of being planted by farmers continues to increase and to extend to over 30 different crops and 30 different developers that are now producing these kind of technology in plants. So from my perspective, I mean, the risks to the environment have been assessed, and they clearly, there've been no incidents that have been recorded over 30 years to date either.
Yeah. Okay. I think we are running out of time, but I'll take a couple of more questions. So it's quite similar question, but it says if the genome edit crop in question is a grass, like ryegrass or turf grass, should the developer first obtain confirmation from USDA that it is non-GMO, and then after development conduct field testing to confirm through the EPA that there are no pesticidal substance? Yeah.
I think when you're making your modification, you kind of know what your target is, right? And what the intention is. So if your intention is to create a pesticidal substance, then you can do both at the same time. In the US, you don't have, it's not a sequential. You can, and then the other thing that's kind of special about the US is you can actually call your regulator in advance and says, I want to do this, like how should I best do this? And they will provide advice to you. So that's slightly different from some countries where it's very hard to have that pre-meeting, if you will, just to make sure that you're in compliance. But in the US, and many times they approach all three agencies, especially EPA and USDA at the same time. FDA, they usually wait until they're closer to the commercial pipeline. That's why you see such a big differences in the numbers. But yeah, you can do both at the same time.
There
And
are
that's why
few
it's
questions
important when you're
about
implementing, sorry, Savannah, it's important when you're implementing new regulatory frameworks. You think about timelines. If you do have to get an assessment that you can be given a quick one, have that pre-assessment, get a determination for, like in the Philippines, for a certificate of non-coverage in 30 days or so that you can make those commercial or, investment decisions early on if you do have to go through that process.
Yeah, there are some questions that I think talk about GM, so I'll skip those. Last question to you, Michael. Can you give an example to what it means by the new arrangement of DNA from crossable species? I think you mentioned in your presentation.
I think what they're talking about is the Fazan's definition of novel DNA there, when I think they're covering certain cisgenesis, for example. If you can bring a gene from a that's a crossable disease, a crossable species. You can bring a gene that may be in a wild type species of tomato into your tomato through CRISPR by using the scissors, cutting it open, and introducing that DNA. You can do that through conventional breeding as well. So they're saying that's not GM food. I think what they're trying to capture is aspects where you change that gene a little bit and you introduce a whole new concept that doesn't exist previously. I think that's the kind of thing, but again, this needs to be worked out as we go through the system as well. But I think the intragenesis part, which we would say still can occur naturally as well, but I think they're sort of trying to consider where you're creating a new concept that doesn't really exist at all, and how they will interpret that. They're producing guidelines now to just actually provide clarity on what they want for that, which will be good for industry. But I think that's the kind of thing they were trying to cover through that definition. And you'll see that in the novel DNA definition that I've included in the slide deck as well.
Yeah, I think there are still question coming up. But I think we have to stop. The people can write to us, and we'll send the question across to the speakers and get back to the question again later on. So I guess we already 20 minutes over time. Thank you so much, Fanley and Michael, for your very informative presentation. And I think all of us benefited a lot. There are more than 200 people who have listened to you. So I guess it's very useful, and we will send a survey to the participants on the future topics of presentation that they would like to hear from us. Again, thank you so much to all, and have a good day. Bye-bye.
Okay, thank you, Sandra.
Thank you. Bye-bye.